Today we're going to have a recap of our third class, but since we have some new roundtable members who are really just getting started with the database, I figured I'd take a minute to show you how to quickly get into the section on the gut immune axis. So when you load the database, this is the opening screen with instructions on the right, and you'll notice on the left where it says menus. I'll show you what happens when you click on that in a second, but on the right, you can see there's another way to find the menus. And let me scroll down a little bit. The top bar has a little tiny icon. Here it is blown up, and that's for pins. So the menu is also pinned up there. So you can always go to the top bar, click on that, and you'll be able to find the menus. So if we click on menus, you'll see one that says Anatomy Physiology. We want Functional Anatomy. And we're going to go to the Body Systems. Then the Digestive System menu. And over on the right, let me scroll up a little bit here, you'll see Gut Immune Axis. So let's click on that. And here we are. Now, today we're going to focus on the mucosal immune system. You'll see that attached on the left side of the screen, the mucosal immune system. So let's click on that and get started with our recap. Our mucosal immune system is the largest component of the entire immune system. It's evolved to provide protection at the main sites of infectious threat, the mucosa. Clinical investigations and in vitro studies indicate that vitamin A is the main regulator of mucosal immunity and could affect immune responses to mucosal infections. So if we look at this illustration, we see we have a lot of mucosal tissues throughout the body. When we're talking about mucosal immunity, it's not just the gut, it's not just the respiratory system, but all of these highlighted colored tissues. These tissues are all sites of communication, where material and information are passed between the body and its environment because of their physiological functions of gas exchange, food absorption, sensory activity, reproduction. The mucosal surfaces are, by necessity, dynamic, thin, permeable barriers to the interior of the body. This fragility, combined with the vital functions of mucosa, has driven the evolution of specialized mechanisms for their defense. Okay, let's scroll down past some of these videos that are worth watching, and let's talk about the structure and components. It has mucosal barriers. We've been talking about mucosal barriers already. We will be talking about those for a while. They're very important. And you'll see here, if you want to go directly to intestinal mucus, you can click on this hyperlink, or the airway mucus. It has mucosal-associated lymphoid tissue. We call that MALT. And that includes the tonsils, the Peyer's patches in the small intestine, the appendix. It's rich in immune cells and crucial for initiating immune responses. It also contains secretory immunoglobulin A, and we'll talk about that today. That's predominantly found in the mucosal secretions, and it plays a vital role in neutralizing pathogens and preventing their attachment to epithelial cells. It also contains innate immune cells and adaptive immune cells. And so what are the functions of our mucosal immune system? Well, it's critical for pathogen defense. It provides a barrier against pathogens It also initiates immune responses to get rid of them. It's also for immune tolerance. And what that means is that we've recognized that something is useful, beneficial, friendly, and we do not want an immune response against it. This is that amazing process where the body can distinguish between friend and foe. It's involved in homeostasis. It helps maintain a balance between immune activation and tolerance, ensuring that the immune system doesn't overreact to non-threatening antigens. If we scroll down a bit, there's a good diagram of the mucosal immune system in the gut wall. We've talked about some of this already. Here are the villi in the small intestine, and the gut lumen is up here. These are the epithelial tissues in the villi. It's filled in with the lamina propria. We see the crypt down here where the stem cells exist to replace the epithelial tissue that dies off pretty quickly. Every 4 to 5 days we get a new set of epithelial tissue in these villi. That's also where the Paneth cells are. Now here we see the Peyer's patch, and it has M cells in it. We haven't talked about those yet. Follicle-associated epithelium, isolated lymphoid follicle right here, and this is just showing the drainage through the villi into the lymphatic system, and it goes to the mesenteric lymph node. We'll cover that later. Now let's talk about mucus. Over on the left side of the screen, you find it easily. Let's click on that. As you probably know, mucus is a slippery, gelatinous substance produced by mucous membranes in the body. It plays a crucial role in maintaining our health by protecting and lubricating various parts of the body. While often associated with illness, it's essential for normal bodily functions and serves as a first line of defense against pathogens. So that would make it part of the innate immune system. What's it composed of? Primarily water, which is why we need to stay hydrated. It also contains glycoproteins. Now, glyco refers to a carbohydrate or sugar, and you know what proteins are. So glycoproteins are proteins that have carbohydrate molecules attached to them. There are many different kinds. It contains enzymes, antibodies, and salts. The glycoproteins are the mucins, and those give mucus its viscous and sticky properties. All these components work together to trap dust, microbes, and other particles, preventing them from entering the body and causing infections. The amount of water influences how thick or thin it is. As far as its functions go, well, we've already said protection. It acts as a barrier trapping pathogens, preventing them from reaching the cells. Lubrication. It keeps tissues moist, reducing friction and facilitating the movement of food through the digestive tract and air through the respiratory system. Immune defense. It contains antibodies and enzymes that help neutralize harmful microorganisms. Hydration. It helps maintain moisture in tissues, preventing them from drying out and becoming damaged. Let's go to intestinal mucus. On the left, you'll see mucin there, and we'll talk about that in a minute. That's one of our tiles. And all of these substances here have an effect, a beneficial effect, on our intestinal mucus. Over on the right-hand side, we have another diagram that illustrates how the mucus layer is a bit different between the small intestine and the large intestine. This diagram shows the thickness, and at the bottom you see it says small intestine, and we see that it thins out around the 300 mark. We've got it through the duodenum, the jejunum, and the ileum. Those are all parts of the small intestine. But when we get down to the colon, the large intestine, there's a lot more mucus. There's an inner layer that's quite thick and an outer layer that has less linking. And we have a closer view of this. So the small intestine effector sites. Effector just means those are usually immune cells, but any cells that have an effect on something else. So again, you see all of these epithelial cells here in the small intestine. There's the crypt, there's the lamina propria, and this green color shows mucus. It goes all the way down to the crypt. We can see commensal bacteria up here at the top, and that's where they kind of make a home and a biofilm to live in. We've got these little things are called secretory immunoglobulin A, or IgA. We'll talk about that in a minute. So they're floating around. Some of them are floating around in this mucus. And over on the right side, we see what's going on in the large intestine. And look at how much thicker the mucus is. It's a darker green. And it's really hunkering down over those epithelial cells. And there's still more mucus on the top, the outer mucus layer. That part of the mucus is flowing, and it's taking things out of the digestive tract and eliminating them. This inner mucus layer is quite thick, and it doesn't move so much, and it's really there as a protective barrier. The intestinal mucus has electrolytes, and those are the salts. It has lipids, and those contribute to the hydrophobic barrier. So it does all of the same things that we've already talked about, but it also has what are called antimicrobial peptides, AMPs. You can click on all of these to find out what they do. They're retained in the intestinal mucus layer without losing their efficacy, and they play crucial roles in maintaining intestinal homeostasis, preventing bacterial adherence and translocation, and protecting against infections. The mucus layer acts as a reservoir for these AMPs, concentrating them to effectively kill bacteria while reducing pressure on the luminal commensal bacteria. Now, that just means our beneficial gut microbiome to require resistance. As far as nutrient absorption, while it's primarily a protective layer, the mucus layer also plays a role in nutrient absorption. It helps maintain the appropriate environment for the absorption of nutrients by regulating the pH and the ionic conditions at the epithelial surface. As far as significance in health and disease, health implications, well, a healthy mucus layer supports a balanced gut microbiome, and we know how important that is. And that is essential for digestion, nutrient absorption, and immune function. And of course, the barrier function. A compromised mucus barrier can lead to increased intestinal permeability, leaky gut, and that's associated with inflammation and autoimmune diseases. As far as its disease associations, it's linked to inflammatory bowel disease, Things like Crohn's disease, ulcerative colitis are linked to alterations in mucus composition and function, leading to chronic inflammation. Infections. A weakened mucus barrier can increase susceptibility to infections by pathogens, such as bacteria, viruses, and parasites, and then irritable bowel syndrome. Changes in mucus secretion and composition can contribute to symptoms of IBS, including abdominal pain and altered bowel habits. So, what factors affect mucus production? First of all, diet, of course. Food is medicine. Dietary components, such as fiber, impact mucus production. A diet rich in fiber promotes the growth of beneficial bacteria that support mucus integrity. However, a diet high in processed foods and low in fiber negatively affects mucus production. We come back to hydration. Adequate hydration is essential to maintain the water content. Stress, we talked about that last week. Chronic stress can alter mucus production and composition, potentially compromising the protective barrier and leading to gastrointestinal issues. And then medications, things like NSAIDs. There we're talking about that Tylenol, we're talking about aspirin, and many others. And then antibiotics, those can affect mucus production and integrity. Now in energetic herbalism, especially TCM, Chinese medicine, we have something called intestines mucus damp, and that kind of fits in here. That would manifest as appetite loss, lethargy, abdominal distension and gurgling, nausea, a sticky sweet taste in the mouth, and alternating diarrhea and constipation. The root of these cold digestive disorders is often an intestinal dysbiosis. Remember, dysbiosis means imbalanced gut microflora that encourages overproduction of intestinal mucus and toxic microbial byproducts. So what herbs have a beneficial effect on intestinal mucus? There are quite a few, but these highlight some of the best. Slippery elm, marshmallow root. We did talk about marshmallow root briefly, and we'll do that in this video. Aloe vera, chamomile, deglycerized licorice. Now we've got marshmallow root, chamomile, and deglycerized licorice in our tiles for the round table. These herbs work in various ways to support the intestinal mucus layer, soothe inflammation, and promote overall gut health. And then you can look again over here and see other herbs that assist. When you have time, you might take a look. In fact, we probably will in a future class at this section about intestinal mucus barrier deterioration. Okay, let's look at mucin. That's one of our tiles. That's a glycoprotein, as I already mentioned, and it's a major component of mucus, found in human and animal secretions and tissues, such as saliva, the lining of the stomach and skin. It's a major component of the mucus layer, protecting the epithelium of the gastrointestinal tract from damage. Mucin also lubricates epithelial cell surfaces and is involved in modulating immune response, inflammation, adhesion, and tumorigenesis. It's the primary gel-forming component of mucus. Unregulated mucin production can affect the health of the host. Deregulation of mucin production is a link between inflammation and cancer. Let's talk about where they're located in relation to the epithelial layer. So there's two main areas. There are secreted mucins, and those form a mucus layer covering the epithelial cell. In the small intestine, there's a single, discontinuous, impenetrable mucus layer. In the large intestine, there are two distinct layers. We've seen that already. In the large intestine, we've got that inner layer, and that outer layer. And there are also transmembrane mucins. Those are attached to the cell membrane of our enterocytes, covering their apical surface. Remember, apical means the top part. And they form part of the glycocalyx, which is a carbohydrate-rich layer directly on the epithelial cell surface. And if you look on the left, you'll see we've got a bunch of different types of mucins. I'll leave it to you to come in the database and look those up if you're interested. Here's a good diagram showing the small intestine, and we see on the far left here what a transmembrane mucin looks like, and there are several of them that are transmembranes. And so at the top part you see the mucin, and here is the membrane of the epithelial cell, and there's a little opening there where it stuck its tail down inside. And what's going on here is a communication. Now remember, we talked about the AMPs, and that's shown here with these little dots. There's the crypt again. Here we've got the epithelial cells. Now that glycocalyx is shown here in dark green. That's a very thin, thick layer that just is attached directly to the epithelial cells of the villi. And if we look down at the bottom, we've got that crypt again with the Paneth cells. We see our goblet cells. Over on the right side of the diagram, we see the single loose mucus layer that has the gel-forming mucins in it. At the top, we've got the lumen with all different kinds of bacteria. We see how some of them drift down into the mucus where they're going to get caught and dealt with. In the large intestine, it's a little bit different. We've got different type of cell. Those are called colonocytes. The epithelial cell is called a colonocyte in the colon. And here's that dense inner mucus layer. It has its glycocalyx again, and that loose outer layer. It also has those transmembrane mucins. So how do transmembrane mucins differ from secreted gel-forming mucins? First of all, they're anchored. The transmembrane mucins are anchored to the epithelial cell membrane. They have that little tail for intracellular signaling. And the extracellular, that's the part outside of the cell, has domains that are written something called glycosylated tandem repeats that form part of the glycocalyx. As far as the secreted ones, those mucins don't have a transmembrane domain. Instead, they're secreted into the lumen by those goblet cells. We talked about goblet cells already. And they form a hydrated gel-like mucus layer. As far as their location, we already talked about that. As far as their functions go, the transmembrane mucins, they form the glycocalyx to protect epithelial surfaces. They participate in cell signaling through that cytoplasmic tail, and they provide steric hindrance to prevent pathogen adhesion. The secreted ones form a lubricating and protective mucus gel that traps pathogens and debris. So what about mucins as a therapy? We know that bacteria in our gut should not come into direct contact with the epithelial tissue. They embed in the mucus by these single or double-layered mucins, which have different genetic compositions. Mucins have missed out as a therapeutic tool over the last few years because people have concentrated more on other aspects. But it's clear that mucin production, mucin quality, and to that extent mucin density is an important part of our immunological defense. It is not just a benign goop, but as a way in which we allow ourselves to communicate effectively between the bacteria and these epithelial cells. Epithelial cells aren't simply barrier cells, but they're also immune receivers and immune translators. So if we have, say, a typical patient present with a functional gut problem, and one of the characteristics is episodic diarrhea, which may include transit mucins present as a white stringy mass in their stool samples, not bleeding, but just white stringing. There's a temptation to say, oh, I've got too much mucus. Well, mucus is both a defense mechanism. It's pumped out, which is an expensive exercise that takes a lot of energy to do. It takes a lot of energy to wash it away. And that's probably the way most clinicians see this, as a mechanism for facilitating an eradication. Whereas, if you have a healthy composition of certain types of bacteria, which can best be amplified by a food selection rather than oral ingestion, then they communicate with the epithelial cells, the Paneth cells, the goblet cells, to produce lovely, healthy, fresh mucins, which in turn means that we can induce tolerance. That means no food allergies, for example, and a collective bacterial composition that favors metabolic fitness and microbial diversity. And food selection is the primary way we can favor Akkermansia municiphala. Now we'll get into that one in just a minute. That's the best, most understood microbe to stimulate mucin production. We know how to do this. And that's by encouraging patients to choose the right type of food and the right type of soluble or insoluble fiber that favors the organisms necessary to break down mucins as they become aged, and to stimulate goblet cells to manufacture more. We also use certain monosaccharides or polysaccharides to influence these mucins, things like mannose, neuraminic acid, and fucose. Those are all hyperlinked, so you can click on them and see what they are. All of these monosaccharides gained notoriety around a decade ago, but they've sort of fallen by the wayside. Fundamentally, we're looking to favor food groups that contain different aspects of fibrous constitution, which the acidic environment of our colon relishes the opportunity to dismember those foods and utilizes them not only for their own self-reproduction, but for sophisticated immune-enhancing components. Once we recognize that the decision about what we place on the fork and plate, then in our mouths, reflects 30 feet later in a very complex tube, as to how that translates over months and weeks and years into health and recovery from illness. Now, you can scroll down here and learn more about how mucin modulates the immune response, and we'll see also details on digestive orders that do involve excess mucin. Those are cystic fibrosis, irritable bowel syndrome, ulcerative colitis, Crohn's, celiac, something called microscopic colitis, diverticulitis. So I will leave it to you to read the rest of this. Now I think it's time for us to take a look at Akkermansia municiphala, another one of our tiles. And you will find that right down here. Let's click on Akkermansia. A couple of good videos to watch. One general explanation of it, and here we see research that shows it could alleviate ulcerative colitis. So it's a mucin-degrading bacterium, and its abundance in our intestinal tract is inversely correlated to several disease states. It resides in the mucus layer of the large intestine, where it's involved in maintaining intestinal integrity. There is a little bit also in the small intestine. It has really beneficial action in metabolic diseases, and it fortifies the barrier function. It's associated with a decrease in inflammation, obesity, diabetes, and metabolic disorders. However, excessive colonization by Akkermansia can break the balance between secretion and degradation, and that can damage the intestinal barrier, leading to colitis. So sometimes folks reach out for a probiotic of Akkermansia, and it's recommended that before you buy a supplement of Akkermansia, you get tested to make sure that you aren't overdoing it. You can increase your levels easily by eating polyphenol-rich foods like onions, garlic, leeks, artichoke, yams, agave, bananas, pomegranate, cranberry, green tea, acacia fiber, Brussels sprouts, okra, cauliflower, broccoli, and chicory root. You can also take an inulin supplement. You can boost your levels by fasting because it feeds off your mucus, not your food. So let's take a look at some of the dietary substances more closely that help maintain a good, healthy level of Akkermansia. As far as prebiotics that it likes those fructooligosaccharides, FOS. Those are short-chain carbohydrates that it can easily ferment, and that will stimulate its growth and activity. Those you can get from chicory root, Jerusalem artichoke, and some fruits like bananas, and there we're talking about the greener bananas and asparagus. It likes inulin. That's a prebiotic fiber. You can get that in wheat, onions, garlic, and Jerusalem artichoke, and it's available as a tasteless powder that you can add to your food. And then as far as polyphenols go, certain polyphenols, like the ones that we find in green tea, red wine, berries, and dark chocolate, may act as prebiotics indirectly by promoting the growth of other gut bacteria that stimulate Akkermansia. As far as plant-based extracts, it likes blackberry seed powder extract, pomegranate peel extract. That's rich in ellagic acid, which has positive effects on Akkermansia growth. And if you eat pomegranate, save that peel and tincture that peel to make your own extract. Urolithin A, and that is a gut metabolite of the elegotannins. It can boost Akkermansia levels. And then we have berberine. That promotes Akkermansia growth by stimulating mucin secretion in the colon. Other useful dietary components include omega-3 fatty acids and capsaicin. That helps indirectly by promoting mucin production. Remember, mucin is a preferred food source for Akkermansia. Important points to remember: individual variations in gut microbiome composition and other factors can influence the effectiveness of these dietary substances. And some food sources can have side effects in certain individuals. So always start with small amounts and adjust gradually. Now let's take a closer look at berberine. Berberine has a significant positive effect on Akkermansia in the gut. You can get berberine as a supplement. Start with 500 milligrams a day. So we see an increased abundance of Akkermansia. There's indirect promotion. It acts as a prebiotic, indirectly promoting its growth by stimulating mucin secretion. Its mechanism: berberine increases intestinal expression of tight junction proteins and the thickness of the colonic mucus layer. That helps restore gut barrier integrity. As far as its metabolic benefits, the increase in Akkermansia abundance associated with improvements in high-fat diet induced atherosclerosis, metabolic endotoxemia, and reduced arterial and intestinal inflammation. It is dose-dependent. So how does degradation of mucin by Akkermansia benefit the mucus layer? Well, maintenance. Akkermansia uses mucus as its sole carbon and nitrogen source, producing mucin-degrading enzymes, and that process helps maintain the mucus layer and contributes to gastrointestinal tract homeostasis. Akkermansia accounts for between 1 and 5% of the fecal microbial composition in healthy adults. It's also involved in nutrient cycling. By degrading mucin, it releases mucin-glycan oligosaccharides that support growth of other beneficial bacteria, and that helps maintain a balanced gut ecosystem. Structuring the mucus layer. The mucus layer in the intestine is divided into two layers, as we saw, and we're talking about the colon here. A dense inner layer that serves as a barrier, and an outer loose layer that creates a niche for our commensal bacteria. Akkermansia's mucin degradation activity helps maintain this structure, allowing it to adhere to mucins and their glycans in the outer layer, strengthening enterocyte integrity. It's been shown to adhere to the intestinal epithelium and strengthen enterocyte monolayer. This interaction contributes to overall health, and it regulates mucus production. The interaction between Akkermansia and the mucus layer appears to play a role in regulating mucus production. It's important to note that while its mucin degradation generally benefits the mucus layer, excessive colonization or consumption of mucin can potentially lead to thinning of the mucus layer and increased inflammation. So we want to keep a balance. Now, I'm not going to go through this just in the interest of our time together, but I want to show you. You can come in here and see exactly what role it plays in regulating our signaling pathways. Some of this is kind of complicated, but all these terms are in the database, so if you want, you can look them up and find out what they are. And here you can learn about how it affects metabolic disorders, including obesity, and how it affects mitochondrial oxidation. That plays a role in colon cancer. So foods and medications or herbs that are harmful to it include antibiotics, and taking a supplement of Akkermansia after you've been on antibiotics can be harmful. It does not like high-fat diets or low-fiber diets. There are certain herbal medicines that could be harmful to it, and those are going to be compounds with really strong antimicrobial properties. For example, oil of oregano and other factors. Post-antibiotic reconstitution. Supplementation of Akkermansia immediately after antibiotic treatment can lead to negative outcomes. And excessive enrichment, taking too much of a probiotic of Akkermansia. There's stuff about animals, and here you can learn about its role in colon cancer. Of course, we've got a lot of studies here that show its effect on allergies, diabetes, its gastrointestinal effects, mental health disorders too. Okay, folks, let's finish up with secretory IgA, immunoglobulin A. So we have regular, plain old ordinary immunoglobulin A that's produced by B cells, and normally the IgA is attached to the B cell. However, the B cell can change into something called a plasma cell, and plasma B cell releases IgA into the lamina propria and other areas, and when it's been released or secreted, it's called secretory IgA, and it has a different structure when it's a secretory IgA. It's a unique feature of the mucosal immune system. So it's secreted into the mucosal layer, not only in the gut, but in the other mucosal areas of the body, such as the respiratory system. And once it's released, it can neutralize toxins before they get in. So let's take a look at this diagram, and we see three different ways that it works. Now, first of all, this little funny looking thing here is the secretory IgA. Actually, let's look at the middle column first, because down at the bottom you see what it looks like before it's attached to anything. Antibodies, which are the same thing as immunoglobulins, are Y-shaped, And in this case, we've got two Y's joined together. Secretary IgA is two of those Y shapes, two of those immunoglobulins joined together. So it's released into the lamina propria, and it can travel up to the surface mucosal layer. And once it's up there, it can bind onto pathogens. We see here two pathogens. Each arm of that IgA is grabbed onto one of those. And it's not actually killing them. What it's doing is preventing them from being able to enter into the cell. And it does the same thing for toxins. So it's grabbing stuff, preventing it from being able to get into the cell. And it's just going to float along that loose layer of mucosa and be excreted at some point. The other thing that it can do is it can, if there's some kind of pathogen that has actually gotten into the cell, or a toxin that's gotten into the cell, it can move up into the cell, grab a hold of it, and then bind it so it can't do anything else. And we then see that it can, on the last panel, export things from the bottom. Let's say toxins or bacteria got all the way down into the lamina propria. Here it's grabbing them, and through channels in the cell, it's moving them up and out, and it's binding to them. It acts as the first line of adaptive humoral immune defense. We haven't really gotten into the adaptive humoral immune system yet. That's coming along. But it's a very important part of our mucosal immunity. And the short-chain fatty acids that are produced by our beneficial gut bacteria promote production of IgA. So that not only protects against pathogens, but it also promotes beneficial bacteria at the same time. And if you come into the database and look at this, you'll learn more about the structure, its function. It has several functions. It neutralizes pathogens, prevents them from adhering to and penetrating epithelial cells. It has immune exclusion by binding to antigens. It facilitates their removal from the body through mucosal secretions, and that could be saliva, tears, and mucus. It has an anti-inflammatory role too, so it helps maintain homeostasis by preventing excessive inflammatory responses that could damage tissues. And it helps to regulate our microbiome. It helps balance the microbiome by selectively binding to commensal bacteria, promoting their retention and growth, while inhibiting pathogenic bacteria. Here's a section explaining how it interacts with pathogens in the gastrointestinal tract. And this section will discuss how it's produced and regulated. I'm going to scroll down to this part, short-chain fatty acids. That short-chain fatty acids are one of our tiles, and they promote both immunoglobulin A and secretory immunoglobulin A, both of them. And so let's see what else we want to talk about today. You can learn about the mechanisms and all that. We get back to vitamin A again. So remember, vitamin A is the vitamin for the mucosal immune system, and it influences the secretion of the secretory IgA in a lot of ways. It's essential for transporting and secreting it across the mucosa. Studies show that vitamin A deficiency adversely affects the secretory component of immunoglobulin A, and it leads to impaired mucosal immunity. Vitamin A deficiency interferes with proliferation and maturation of epithelial cells. It decreases the activity of brush border enzymes, which we covered in a previous video. I'm going to skip some of this that we haven't covered yet. It'll be confusing to you. It helps regulate immune cell populations. it is involved in imprinting gut homing properties. So what that means is there are different mechanisms that help various immune cells home in on an area of concern. It's like a homing pigeon. We have methods where we can call out for help, and in the gut, it helps to set that whole process in action. And vitamin A enhances the secretory IgA levels, and vitamin A regulates mucin expression. So we see vitamin A as a big player in a number of these mechanisms. As far as the clinical significance of secretory IgA, it's important for infections. If we don't have enough of it, we can have more infections. It helps with autoimmune disease. If that secretory IgA production is dysregulated, its lack of function can contribute to development of autoimmune diseases. It also plays a role in allergies. It can help modulate allergic responses by preventing allergen penetration. It also promotes tolerance. And it's a diagnostic marker. So as far as dietary components that affect it, first of all, dietary protein. So high protein diets increase secretory IgA production in the gut. It also is good to include those dietary polysaccharides because they can enhance its production by inducing specific immune responses in the gut. For example, polysaccharides can stimulate production of IgA specific to their structures, contributing to microbiota regulation and immune tolerance. Here we've got a section on how does interaction with commensal and pathogenic microbes enhance its production. I'm going to leave it to you to read that part. So I think we've covered a lot in this video. We will have much more to follow in the next one, so I hope to see you guys in class. And for those of you who are unable to attend class, hopefully you are going to find that these videos are providing you with a good foundation. And I would encourage you to subscribe to the database so you can dig into it all further. With that, I say have a good day, and I will see you next time.